Welcome to This Explainer. Have you ever looked out at the crowd during a massive, packed soccer match? I mean, really looked? If you scan the stands, you'll see just the most marvelous variety of humankind. We're talking different eye colors, hair textures, heights, builds, the whole shebang. But what are the underlying rules that dictate how all these incredible variations are actually passed down from parents to their kids? Well, today we're going to unpack the foundational rules of genetics. We are taking a journey from a quiet Abbey garden all the way to the cutting edge of human genetic testing.
Okay, let's dive into this. Back in the 1800s, the leading assumption was something called the blending hypothesis. People basically thought genetic material from two parents just mixed together, the same way you'd mix blue and yellow paint in a bucket to get green. But think about the huge logical flaw there. If that were actually true, over many generations, everyone would just blend into one uniform appearance. And we obviously don't see that, right? Plus, the whole paint theory totally fails to explain how a trait can completely disappear in one generation, only to magically pop back up in the next.
To solve this puzzle, here's our roadmap. 1. The mystery of hereditary. 2. Mendel's garden rules. 3. When genetics gets complex. 4. Tracing human traits. And finally, 5. Genetic disorders and testing.
Starting off with part 1, the mystery of heredity.
So how did we first actually begin to understand the transmission of these heritable variations? To replace that flawed blending idea, that pail of paint analogy where traits just irrevocably dilute and mix, scientists needed a completely new model. Enter the particulate hypothesis. Instead of mixing paint, think of this more like shuffling a deck of cards. In this model, parents pass on discrete, individual heritable units, which we now know as genes. Just like playing cards, these genes can be shuffled and dealt out generation after generation while completely retaining their separate, undiluted identities.
Moving on to part 2, Mendel's garden rules. The very first person to document this shuffled deck mechanism was a monk named Gregor Mendel. And get this, he did it decades before anyone had ever even seen a chromosome under a microscope. Mendel chose to work with garden peas because he could strictly and meticulously control their mating. Here's exactly how he pulled it off. First, he'd remove the pollen-producing stamens from a purple flower so it couldn't self-pollinate. Second, he'd transfer pollen from a white flower directly to that purple one. Third, he waited for the pod to mature, and finally, he planted those specific seeds. Because of this strict methodology, he was always 100% sure of the parentage of his new plants. He was crossing true breeding varieties just to see what would happen. Now, what's really interesting about this slide is what happened next. When Mendel crossed those purple and white flowers, the first generation of hybrids were all purple. The white trait seemed entirely lost. But then, he let those purple hybrids self-pollinate, and the math revealed an absolute smoking gun. Out of that next generation, he counted exactly 705 purple flowers and 224 white flowers. That is a mathematically precise 3-to-1 ratio. It proved beyond a doubt that the heritable factor for white flowers wasn't destroyed or blended away, it was literally just masked by the purple factor. From this, Mendel realized there must be alternative versions of genes, which we now call alleles. Take the gene for flower color, for example. It exists in 2 versions, one for purple and one for white. Every organism inherits 2 copies of a gene, one from each parent. If those 2 alleles differ, the dominant one dictates the appearance, and the recessive one, it stays hidden. During reproduction, these 2 alleles separate, or segregate, meaning a sperm or egg only gets one of them. This brilliant piece of deduction is what we now call the law of segregation. Because dominant alleles can completely hide recessive ones, what you see isn't always what you get genetically. And this introduces 2 absolutely vital terms. Phenotype is the organism's observable trait, like actually having purple flowers. Genotype, on the other hand, is the underlying genetic makeup. A purple flower could have a genotype of 2 dominant purple alleles, or it could have 1 dominant purple and 1 hidden recessive white allele. Same phenotype, entirely different genotypes. And Mendel didn't just stop at flower color. He observed the exact same mathematical pattern across multiple traits: seed color, seed shape, pod shape. Whether he was looking at yellow versus green seeds or round versus wrinkled seeds, that second generation constantly hovered right around that exact 3-to-1 ratio. This proved his law of segregation wasn't just some one-off fluke. It was a fundamental mathematical rule of nature. Which brings us to part 3, when genetics gets complex. As we know, nature absolutely loves to throw us a curveball. What happens when we introduce real-world genetic complexities? Mendel wondered, what if we track two traits at once, say seed color and seed shape? Do they stick together like a package deal, or are they inherited independently? Well, by crossing plants that were heterozygous for both traits, he got a brand new ratio, 9 to 3 to 3 to 1. 9 yellow round, 3 green round, 3 yellow wrinkled, and 1 green wrinkled. And this brilliantly illustrates his second major breakthrough, the law of independent assortment. It proved that different genes, like color and shape, are packaged into gametes completely independently of one another. But it turns out, dominance isn't always an all-or-nothing game. Sometimes you get what's called incomplete dominance, where the traits actually do seem to meet in the middle. Like if you cross red and white snapdragon flowers, you get pink ones. The alleles themselves don't blend, but the heterozygote produces an intermediate amount of pigment. Then you've got codominance, where both alleles are fully expressed side by side. If you have the human AB blood type, your red blood cells prominently feature both A and B molecules. Neither one subdues the other. And it gets even more intertwined from there. Sometimes one gene completely alters the expression of another. That's a process called epistasis. For instance, in Labrador retrievers, one gene determines the color, but a totally separate gene decides like that pigment ever even makes it into the hair. Then we have polygenic inheritance, where multiple different genes additively affect a single trait to create a continuum. Think about human height or skin color. And finally, we absolutely can't forget the environment. Things like sun exposure, nutrition, and lifestyle interact directly with your genotype to shape your final observable phenotype. It's the ultimate combination of nature and nurture working together.
Let's look at part 4, tracing human traits.
So how do we study all this in humans? Since we obviously cannot, and ethically should not, breed humans in controlled experiments like pea plants, geneticists have to act as historical detectives. By tracing phenotypes back through family tree pedigrees, we can actually deduce genotypes. Take a widow's peak, that pointed hairline. Pedigree analysis shows us this is driven by a dominant allele. Conversely, the inability to taste a bitter chemical called PTC is a recessive trait. By mapping out parents and grandparents, we can use Mendelian probability to calculate the exact mathematical odds that a future child will inherit these completely innocuous, everyday variations. So, the crucial point is this. What happens when the trait we're tracing isn't just a hairline, but a severe genetic disorder? This brings us to the concept of a carrier. A carrier is someone who has one normal dominant allele and one disease-causing recessive allele. Phenotypically, they look and feel totally normal, but genotypically, they are carrying a hidden, dangerous card in their genetic deck that they can pass down to the next generation completely unnoticed.
And that leads us to part 5, genetic disorders and testing.
Here is where the math of Mendel's garden meets the real-world gravity of human healthcare. Genetic disorders generally fall into two categories. Recessive disorders, like cystic fibrosis, require a child to inherit two recessive alleles, one from each carrier parent, for the disease to actually manifest. Dominant disorders, however, require only one single copy of the allele to cause the condition. Now, lethal dominant alleles are incredibly rare, mostly because they usually cause death before the person can reproduce. But exceptions do exist, like Huntington's disease. It's a fatal dominant disorder that hides in the genome and doesn't strike until a person is in their late 30s or 40s, which is often after they've already had children. Sometimes genetics presents us with a fascinating and frankly awe-inspiring evolutionary compromise. Take sickle cell disease. It's a recessive disorder causing severe blood flow issues. But if a person is just a carrier, meaning they have the sickle cell trait with only one copy of the allele, they are usually totally healthy. Even more remarkably, having this one abnormal allele actually reduces the severity of malaria attacks. Therefore, in tropical regions where malaria is highly prevalent, being a carrier offers a distinct evolutionary survival advantage. It perfectly explains why this allele still persists in certain populations today. Thankfully, today, we don't have to guess our odds in the dark. Modern genetic counseling relies heavily on Mendel's probability laws. Prospective parents can undergo carrier identification blood tests to see if they hold recessive alleles for diseases like Tay-Sachs or cystic fibrosis. If they're pregnant, fetal testing techniques like amniocentesis or CVS can actually analyze the DNA of the developing baby. And immediately after birth, routine newborn screening can detect certain metabolic disorders like PKU early enough to treat them effectively with simple dietary changes. Let's move to and see how this builds into our final takeaway. We've moved from the simple probability of pea plants to the incredible, almost sci-fi ability to read our own human source code. But as genetic testing reveals the exact cards we hold in our deck, it leaves us with immense, heavy choices.
It's a huge question to ponder. Do you want to know what diseases you might develop later in life? How does having this kind of knowledge shape the decisions you make about your own health or about starting a family? Mendel's simple garden rules open the door to the ultimate understanding of human biology, but it is entirely up to us to decide how we navigate the complex future it reveals. Thanks for joining me for this explainer, and keep learning.